Today we're talking about lactoferrin concept. The operon is a set of positive genes on the same stand in a genome that are co-transcribed into female foreign central. So especially in bacteria, the genome consists many operon models, for example, lactoferrin, tryptophan, etc. So these structural genes are present in a polysystonic form, and they are under the control of single promoter. The lab operon is a operon model where the genes are coded for different enzymes which controls or which are involved in lactose metabolism. The main components of lab operon includes the promoter region, operator region, and three structural genes called lacto. The promoter region is the sequence where the RNA polymerase can bind and initiate the transcription process. So there are separate names for LAC and gene. The LACer is the gene which goes for the repressor protein, and these repressors are continuously synthesized under the control of LACer gene. And there is a separate promoter for LAC, Z, Y, and A genes. The operator sequence, this is the binding site for repressor molecules, basically overlaps with the promoter of structural genes. Then we have a downstream lateral gene, which goes for the repressor molecule. The structural genes like LAGZ goes for beta-galactosidase, which helps in the cleavage of the disaccharide lactose into monosaccharides like galactose and lactose. LAC-Y gene includes the lactose proteases. It is a membrane protein that facilitates uptake of lactose from the surrounding of the bacterial environment. Then the LAC-A gene, which includes the beta-galactoside transaminase. Okay, the main function of this enzyme is not at completely understood, but it is said to be involved in metabolism of lactose. Okay, there will be two situations when there is a glucose is present in the environment around the bacterial colonies. So they take up glucose as a primary source of energy. So when there is an absence of lactose and presence of glucose, the lactoferrin must be switched to GS non-function. So what happens here? In the absence of lactase, there is a repressor molecule. The repressor molecule is produced by LAC-I gene, and that is controlled by LAC-I gene. So once there is a presence of repressor molecule, in the absence of lactase, it binds to the operator region. Okay. So once the binding of repressor takes place, the RNA polymerase cannot bind to the promoter without transcription. If there is no transcription, no functional proteins are synthesized. So in the another case, there is absence of glucose and lactose is available as the secondary source of energy for the bacterial cells. So now what happens? To metabolize these lactose, the lactose production gets switched on. So now the lactose or its isoform allolactose will bind to the receptor molecule. So now the promoter and operator regions are available for the binding of RNA polymerase. So basically RNA polymerase bind to promoter region. So once the promoter region is bound by RNA polymerase, it brings about transcription by synthesizing a polysystonic. So once the mRNA is synthesized, the translation takes place by synthesizing the different proteins or the enzymes which are essential. in the bacterial cell. So this is how the presence of lactose will regulate the lack functional genes. The lactose is mainly controlled by presence or absence of lactose. So this can be given as an example for positive as well as negative mode of gene regulation. Thank you.